welcome to the Weekend Update. I'm your host, Tommy. And I'm Tommy, and here are your top stories. Today, world leaders are shocked to discover that bacteria in human wastewater is actually being decomposed by microscopic organisms, Rotifera. That's right, Tommy. These organisms range between 200 and 500 micrometers long. They were first discovered by
Reverend John Harris in 1696. Oh, that's just my screensaver, sorry. Their taxon contains 2,200 species and is divided into three classes: Mononoganta, Doloidea, and Sisonidea. Composing the largest number of species, Mononoganta accounts for a wide variety of freshwater rotifers and can be found both in free-swimming and sessile forms. Doloidea are unique in that they reproduce exclusively by parthenogenesis and can enter a dormant stage in harsh environments. Sisonidea are found on the gills of marine crustacean known as Nobalia. These organisms have a worm-like structure to do a well-developed external cuticle. As pseudocoelomates, their body cavities are partially lined with mesoderm. These bilaterally symmetric organisms are divided into three parts: the head, trunk, and foot. The head is composed of the corona, a distinctive feature to this taxon, which resembles a wheel. It is ciliated and can be modified so that the cilia has bristles, tufts, or has been lost altogether. The trunk encloses most of the internal organs and forms a major part of the body. The narrow foot projects from the rear of the trunk, giving the impression of a tail.
These organisms are commonly found in freshwater or moist terrestrial environments, but scientists have found that they can exist in stillwater environments too, such as the bottom of lakes, as well as free-flowing water, such as rivers and streams. To discuss this, let's bring in our senior Rotifera locomotion analyst, Dr. Taylor Lovett. Thanks for dropping by today, Dr. Lovett.
My pleasure. I was just passing by.
Not all members of Rotifera have the same method of locomotion. Some are free-swimming and truly planktonic, while others move by inchworming along a substrate, as can be seen here. You can clearly see the retraction of the foot, which contains adhesive glands to stick the surface. When the foot is pushed back out, this propels the organism forward. There are even a few species within the taxon that do not possess means of self-locomotion and are normally immobile. Those species live inside tubes or gelatinous holdfasts that are attached to the substrate. Rotifers contain specialized organ systems and a complete digestive tract that includes both a mouth and anus. Their feeding method relies on the coronal cilia to generate a current to sweep food into their mouth. Their mouth opens into a ciliated pharynx, known as a mastax. It has a powerful muscular wall with tiny calcified jaw-like structures, known as trophy. And no, this isn't the same as trophy wife. The size of these trophy will vary between species and are dependent on their type of diet. For instance, carnivores have forceps to help them bite prey, while suspension feeders will have grinding ridges. Past the mastax is the oesophagus, which opens into the stomach. This is where most of the digestion and absorption of nutrients occurs.
That's fascinating. Now let's bring in our senior Rotifera sexologist, Dr. Sheila Cruz.
Thank you, Tommy. Well, most species will hatch at a mature version of adult rotifer with respective male and female parts. While the males will hardly grow at all, the females will grow up to 10 times the size of their male counterparts. Their lifespan for females varies from 2 days to 3 weeks, while the males being a lot less.
How come males don't live as long? That's not fair.
Well, isn't that how it should be? Just kidding.
It's because the males don't have a completely functional digestive system, so they are short-lived but are sexually fertile from birth, although Sisonidea males are roughly the same size as the females.
In fact, male rotifers contain a single testicle and sperm duct, while the females will contain a more developed reproductive system that includes 1 to 2 ovaries, each with a vitelline gland that supplies their eggs with yolk. As one can see, fertilization on a sexually reproducing species is internal. As mentioned before, Doloid will reproduce exclusively by asexual parthenogenesis. Monogonata alternates between cyclic parthenogenesis and heterogamy. Reproduction by parthenogenesis allows for fast population growth and colonization of new environments.
Wild. So let's discuss how the Rotifera taxon fit with the other known organisms. Using phylogenetic trees.
Our first phylogenetic tree is based on morphological similarities between species. It places Rotifera between Annelida and Echinodermata. Rotifers are often compared to annelids due to their similar characteristics, including a hydrostatic skeleton and respiration through cellular membranes. However, upon 16S RNA sequencing, we found that Rotifera is more closely associated with mollusca, and common characteristics with Annelida are likely resulting from convergent evolution.
Let's step back and look at how these trees relate to previous scientific understanding of where the Rotifera taxon belonged. You know what is so sad about Rotifera? What? That they're teeny? That no one on Earth can even see them? Aside from that, there is an uncertain placement on the phylogenetic tree. The Acanthocephala were considered to be a separate phylum, but they are really modified rotifers. What does that mean? This means that because of the anatomy of the Rotifera and the findings of new species always leads to modifying the phylum and the cladogram. Basically, Rotifera is confined to the Doloidea and the Mononoganta. They have concluded that Rotifera is most closely related to mollusca. Funny that we have the same conclusion given their PhDs. And loads of debt. Good one, Tommy. Anyway, this information is based on cladograms and characteristics of the organisms we studied. Thank you, Dr. Lovett and Dr. Cruz, for your contribution. This has been Weekend Update with your host, Tommy. And I'm Tommy. Have a great evening.